So this is the second part of question 5 on the Foundation Biology end of chapter 4 test. So how much would I get, and what are they looking for in one of these 6-marker practical style questions? So to get 1 or 2 marks, I need to say something about either how the apparatus is set up, how light is supplied, how you can measure photosynthesis, or I might say one thing that needs to be kept the same, a control variable, to get valid results. And if I'm doing that with poor spelling, punctuation and grammar, I might be awarded 1 mark, and if my spelling, punctuation and grammar is better, I might get 2 marks. So to get 3 or 4 marks, I need to tell you something about the setup and something about measuring photosynthesis, or how we can vary the light intensity, or a control variable, or another valid point. And to get it, so if I do that poorly, I get 3 marks. If I do it with better spelling, punctuation and grammar, I get 4 marks. To get 5 or 6 marks, I need to talk about a control variable fully, or mention repeats, and I need to describe how the apparatus is used to measure the rate of photosynthesis at different light intensities given. So, relevant science. You could say placing, in terms of the apparatus, placing the weed in the water, shining the light on the beaker. How you're going to measure the light intensity. So, did you, like me, change the distance that the lamp was from the plant? You could have maybe used layers of A4 paper to get dimmer light intensities, or lower light intensities. That was another way of doing it. How are you going to control the other variables? So you could have said make sure you had the same pondweed, or the same length of pondweed. Like me, you could have talked about keeping the temperature constant, or you could have talked about keeping CO2 constant. So other control variables, making sure that you left sufficient time after changing the light intensities, so you got a reliable reading of your new light intensity. Some information about how you're measuring the rate of photosynthesis, so you could have counted bubbles, or you could have collected the gas in a delivery tube, or you could have used a delivery tube and a syringe to measure the volume of gas of CO2 created, which would be proportional to, it would go up as the rate of photosynthesis went up. So counting the number of bubbles in a set period of time was a way of doing it. And did you mention repeats and taking a mean? So I did, and I hope you did too. So I'm just going to zoom out maybe a little bit. So that is the mark scheme. Feel free to press pause and mark yourself. Alright, see you on question 6.